There are no such things as incurable. There are only things for which man has not found a cure. A very good morning to one and all present here. I welcome you all to our 14th webinar, Product Development for Regulated Markets. I call upon Dr. V. Shankar, Head, Department of Pharmaceutics, to welcome the chief guest.
So, good morning, Bindu Madhavan.
Good morning.
And I welcome all the students and faculty for this webinar. I thank our principal also for organizing this event on a continuous mode on this lockdown period for the benefit of students and faculty of all the faculty in the pharma discipline and the students in the pharma discipline. So just it is my honor to welcome our speaker today, our old alumni. He's Dr. Bindu Madhavan. Now he is the senior manager in Excellent Formulation Development in Chennai. That industry is now currently optimizing the instruments for establishment at the IIT campus. And he has a lot of experience in the formulation development. His biodata or his profile shows he has worked in Sanofi, a legend in pharma field, and also Shasen. He has also worked, and almost he has 10 years of experience in the formulation and development. And he has developed a lot of products with the team. And they have submitted a lot of generic products as well as products to UK, US, all this Australia, all these countries. They have made many immediate release and solid dosage form products for the benefit of human beings. It is highly appreciated, and he is a very active person from the period of studying. So I thank him. Within 2 weeks, he has accepted our invitation, and he told definitely I will discuss the success stories of industries, the students, and what are the qualities the students should have when they want to enter in pharma industry. Really, we are very happy to welcome Mr. Bindu Madhavan for this talk. So I thank him on behalf of PSG College of Pharmacy, principal, management, and teaching faculty for this webinar. And at the end of the session, students can ask their doubts. Any basic doubts also he will clarify, I think. So once again I welcome you, Bindu Madhavan, for this talk. Thank you.
Yeah. Thank you. Good morning, one and all. Actually, first of all, thanks to the organizers of PSG College of Pharmacy. Of course, I studied there for done my M.Pharm there. So very heartful thanks to all of them. They have just given me an opportunity to interact to this webinar with all the students and the faculty members. And whenever I meet or when I talk to Shankar sir, I used to tell him that the students are very good in the theoretical part, but then they were not aware of what's happening in the industry. And whenever I go for any interview, whenever I interview any fresher candidates who are just out of the college, so they know the theoretical part and they are good in their projects, whatever they do. But then they lack the the concept of this product development, actually what's happening in the industry and how they are doing it. So I just wanted to touch on that point. Actually, it's a very broad topic actually. There are a lot of things which are there to discuss, but still I have just confined it certain basics. As a student, if you know what are all the things which I'm going to say to you today, if you know that, it will be useful. So further, you should do a research or you should refer the guideline and you can just study on it. I will also quote some of the guidelines, which is very important for you people, especially for the people who are going to pursue their career, want to pursue their career in the field of research and development. So this is how I framed this presentation, and I'm going to talk on the basics of what product development is and how we are going to do it, and what are all some certain basics requirement. So like that only I have just framed this presentation. So let's get into this presentation. This is what we are going to discuss it today. It's about the product development and how we do it, and what are all the phases. Like product development, it's not a overnight thing, right? It requires a lot of things to be done. So that will be done in a phase manner. So what are all the different phases in general and industry will follow to do a product development, and certain basics of regulatory requirements which has to be considered during our product development, and certain basics of guidelines which we normally follow for this product development. So these are all the things which we are going to be discussing today. So from my suggestion is, students, please make a note of it, and it will be very helpful when you are appearing for any interview, or when not only for that, it will be helpful for your future also. So to start with the product development, I think everybody is aware of this ICH. So there is a guideline called as ICH Q8, which is for the product development, which clearly defines what a pharmaceutical development is. If you read the definition, it says that to design a quality product and its manufacturing process to consistently deliver the intended performance of the product. You know, two words which is very, very important here. One is the quality and the consistently deliver. So from a formulation point of view, or from a formulator point of view, if you see whatever the pharmaceutical product which you or me are going to develop, it should be a quality. You know, ultimately the aim of every formulator should be the end user, or nothing but the general public or the patient. So the quality should be built in within the development. It should be built right from the first day of your formulation development. Not only that, whatever you are developing, the manufacturing process, whatever you are developing, it should consistently deliver. It's not one or two times. So patient, it has to be manufactured continuously over the years. So these two are the key parameters which a formulator should keep in his mind when he is developing a product. Quality, at the same time, the manufacture process, which should be feasible, and it should be consistent across. Like even if you take 10 batches or 15 batches or 100 batches, the quality should not go down and the process should be consistent one. So these two are very, very important from a pharmaceutical development point of view. So next thing is a little definition on the generic. People used to say generic. What's generic is, what we are going to do is, generic product is nothing but comparable to the innovator drug product. This innovator drug product is the one who entered into the market, who got the first marketing authorization first in the market. And it depends on the countries. Like some call it as innovator. In US they call it as a reference listed drug like RLD, and in Europe they call it as a RMP, like reference medicinal product, like that. But basically, a generic product should be in line towards the innovator product in what are all the aspects. That is also very important, with respect to the dosage form, with respect to the strength, with respect to the route of administration, with respect to the quality, efficacy, and the intended use. So this all should be in line with the innovator drug product. Innovator drug product is nothing but a person who got the first manufacturing authorization and is already existing in the market. So we will be developing a product which is in line towards that innovator product. That is what it all about. So I just wanted to just have a definition on this generic product. So moving on to next, which is which is the heart of our presentation, is the product development phases. Like what we do. It's nothing but like your MPharm project only. It's like whenever we decide a project, just consider like I'm going to do this project. The first thing what we do, we will just Google it and we will do a literature search. The same thing happens in industry. So whenever my business development team or my based on the the project comes to me, the first thing I will do is they will say so and so tablet of so and so strength for so and so market. So first thing I will do is do a literature search to understand certain things. So what are all the things that we will do? It's the simple online search regarding the API. API is nothing but your drug or the excipients and what type of formulation it is. So and not only that, we will also have to look into the who is the reference product. Like I told, it's a innovator product and reference product. It's one and the same. So there are various sites which students make a note of it, especially for the US. If you wanted to know about the reference product details, you can refer to Orange Book, Drugs at FDA, and if you want to develop a product for a UK, and it's medicines.co.uk, and for Canada, if you can put it in Google like Canada product search, etc. Like it will give. So depends upon the country of product of interest, or depending upon the country in which we are going to develop, we will search it, and we will know who is the reference product under which we need to do a generic of it. That is very, very important. This is the first step. And also we will search on the monograph with respect to the USP, BP, Farm Europa, various monographs is available, what the API is, and whether the dosage form is official in the monograph. So what is the dissolution media has been given, what is the criticality in it. It is nothing like a simple literature search like how you do for your MPharm project, or in the same way we will do. And with this, we will come to know that yes, this is my reference product. This is how it is. And also we will talk about the patent. We will also look into the patent aspect also. Is there any patent constraint for our generic product development? If an innovator product is a patent protected, and what kind of protection it is, when it is expiring, whether we can able to do the generic product, and how strong the patent is. So all those things we will evaluate at this first stage in itself. And then we will come to a conclusion whether we can able to develop this product, or even if we wanted to develop, what are all the constraints. It's just a theoretical assumptions we will make. Not only once it is done, then we will go for a active. Like obviously, if you wanted to do any product, we need API and excipients, right? So if the API also has to be sourced. It's kind of a raw material which we need to purchase from the outside. So we are not manufacturing the API as such. So in that aspect also, we need to source it, like who's the vendor, whether the vendor is having the approval in that particular market, and at what stage he is. And we will request the certificate of analysis from him, and we will compare it against the respective monograph. If I want a USP grade material, then whether he is really supplying USP grade material, and whether it is up to my requirement and what it is. So it's a first cut evaluation with respect to the active sourcing we will do. Once it is done, then we will also ask him to give some details, like what is his DMF availability, as I told, whether he is complying to a particular monograph, or especially the stage 3, that active, the third point, impurity profile evaluation, whether genotoxic or carcinogenic. It is a very, very important thing now. Like every API will have its own impurity profile. Okay, so we need to evaluate whether that particular impurity is a genotoxic or a carcinogenic. So if that is so, if that particular impurity is a degradant or it is a process driven, so we need to discuss with that particular API vendor, and we should come to a conclusion. That is a very, very important assessment which we should do it at the first stage. And then statement on the non-patent infringement. Like I told you, patent will be on the drugs API part. It can be also in the formulation part. So formulation part, patent, it's up to us that we can deal with that, what kind of patent it is. But when it comes to the patent on a drug substance, that we need to deal it with the particular drug substance vendor, whether he is whatever he is supplying is a non-infringement API, because that is very, very important. Because without that assessment, if we develop and if we file it, then after that, if that particular API vendor is having some issue with some patent litigation and all, then obviously whatever the finished product or the tablet dosage form which we develop also will get into trouble. So at a first stage itself, we should get all those things. And also the commitment to the physical specifications. In some this I would like to emphasize on the particle size distribution. So you people might have studied, and you know that the particle size distribution is very, very important with respect to when it comes to the dissolution of any drug product. So at the first stage, we may not know like what the PSD we should fix. That completely depends on the trials what we are going to try. But still, we should ask that vendor that what are all the different grades or different particle size he's having, and whether we have to procure the different particle size, and we need to do some trials, and then we need to finalize the particle size of the API. So this is how we start. So this is basically a preliminary work, I can say. literature search, who is the API, what is the reference product, what it is. So with this, we can able to understand, we will come to know what product we are going to develop. And then the next is the active testing. So once we are done, yes, this is the source which I want to use. So we will get that API from him for a small quantity, and we will test it here, like physical and chemical testing, in the sense what is the bud density, what is the tap density, and the chemical testing again, like assay and related substance test. It's not like merely we will go with the certificate of analysis whatever he is giving. So we need to test the parameters, and we should ensure that whatever he is giving is the right. And the second thing is the solubility in various pH range. That's very, very important. When it comes to the dissolution, it's completely a solubility based. So not only we have to do it in the various pH range, starting from 0.1 normal HCl, which is nothing but pH 1.2 to 6.8 or sometimes 7.2, like 0.1, 4.5 and 0.1 normal HCl, 4.5 acetate buffer, 6.8 phosphate buffer, and 7.2 phosphate buffer. We should check the solubility of the particular API in these various pHs, and then we will come to know whether the solubility of the API is a pH dependent or an independent, which this details is very, very required when we go for a product development. And also the monograph or the in-house method, like if I'm buying a USP grade material, and whether all the testing method I'm talking about, whether he is following the same as per the US recommended monograph, or he is having any in-house method of his own. If it is in-house method, what it is. So these details we will check it in this part. And then the reference product. In the previous slide, I told you that we will identify the reference product based on those literature search and all those things. So once it is done, in US, it is very clear that they will, if you go to the Orange Book, you can find that if you put a drug X, and all the dosage form which has been listed, it will be listed. And in one column, they will specify it as RLD. That is nothing but reference listed drug. So I prefer students just go and check it in the Orange Book. You can put any drug. So number of drugs which is listed, it will be listed in it. And particularly for some drug which will be there in the brand name, it will be listed as RLD, which is nothing but the reference product which has been suggested by the US FDA. And by default, we should take that as a reference product, and we should procure that particular brand from the market, and we need to do the evaluation. So the sixth stage is that only, the reference product evaluation, which is very, very critical for any formulation, because until unless we are not evaluating that particular, obviously I told that the generic development is nothing but it is in line with the reference. So until we don't know what a reference product how it looks like, it's very difficult for us to develop. So that is the reason why we should procure it, and we should evaluation. This evaluation of reference product is very, very critical. I have mentioned certain things like both physical and chemical evaluation to be performed with respect to what is the tablet weight or the capsule weight, what is the thickness, what is the hardness, what is the DT, how much is the assay, what is the related substance profile it is having. And also, what are all the ingredients? It will be listed in the pill. Like if you go to the patient information leaflet of the particular product, all the inactive ingredients will be listed. So as a generic product developer, we should also, more or less, we should also follow the same inactive ingredients which has been used in the reference product, more or less. Not only that, the dissolution profile, which is very, very important when it comes to the oral solid dosage forms. I have given it as a OGD database within, which is nothing but Office of Generic Drugs, we call OGD. This is for the US market. So you can just type in Google like OGD database or Office of Generic Drug Dissolution database. So once you go into it, if you put that particular drug name, example paracetamol, if you put, and it will suggest what kind of dissolution media you should follow. And for the US market, it is straight away that you should use that particular dissolution in all your testing parameters. So not only that, I have also given it as multimedia dissolution profile, which is helpful. Like we need to predict the drug performance across our pH range. Like if you consider the oral route, it starts with the stomach and it ends in some intestine, or it will go up to colon. So if you do a pH study, like first start with the acidic pH, like pH 1.2, and up to 7.2. You have to do a dissolution profile at various pH, and you should try to understand your product behavior across this. So this is very critical, this reference product evaluation. And once this evaluation is done, and now you will come to know how my reference product is behaving, what it looks like and how it is behaving. So we have to develop a generic product in line with this reference product. So that is the thing. Looks simple, but it's very complex. So this is how it is. And not only that, the container closure system, like how a reference product has been packed, whether it is packed in a bottle or it is a blister, whether it is a bottle, what is the type of the bottle, whether it is a HDPE, whether it is a PP, in the polypropylene or something like that. And also what type of closure they are using, whether it is a child resistant closure or it's a screw type, or what is the thickness of the bottle, like we call it as a wall thickness. What is the overfill volume? Like every bottle will have its own capacity. We call it as a overfill volume. What it is, everything needs to be characterized, and in the same way we need to develop for our, we have to use the same type of container closure system for our generic product. So this is the preliminary phase, which is very, very important, and any miss here in this phase, it will lead to trouble in your development. So you should be very, you should have a thorough understanding of your reference product. Then only you will come to know what you are going to develop and how you are going to develop. So this is what it is. Yeah, at the end of phase 1, now we will come to know what is my target size and shape of any dosage form, what is my weight which I wanted to target, and what is my hardness with respect to the tablet, what is my target dissolution profile which I need to do, what is my packing configuration which I need to follow for a generic development. So once you've done this, you will come to know all these things. And I also given one guideline, students can make a note of it. It's called as the size, shape, and other physical attributes of generic tablets and capsules. It's a guidance to industry which has been provided by US. It clearly talks about the size, shape, and other the physical attributes of your generic product, how it should look like. Example, it clearly says that if your reference product is equal to or greater than 17 mm at any dimension, then your generic product should also be either the same size. Not only the size, you should map in the volume of the tablet. The volume of the tablet you should measure for the reference product, and your generic product should also map with respect to the volume also. So if you are developing a market for US, this is a very critical guideline which clearly defines the size, shape, and other physical attributes of how a generic tablet should be. So it may look simple, like it's a round tablet, I'm also going to give round. No, even a 0.1 or 0.2 mm thickness difference in the reference and your test product will hamper the volume of your tablet, especially when it comes to the tablet which are bigger in size. So if you think overall, all these guidelines, all those things are just made considering the patient compliance. So obviously nobody wants to take a bigger tablet, or even if it's supposed to take a bigger tablet, it should not choke in their throat or something like that. So this is what it is. Any generic development should keep the patient compliance in mind, also the manufacturability with the consistent deliverability in mind. Then only it can be delivered. Of course, it's all defined by our specific guidelines, but still we should ensure it. So this is the end of phase 1, where now we came to know what we are going to do and how we are going to do. Now, this is the phase 2 of this is the heart of any development. Now I know what I'm going to do. Now this says how I'm going to do. Like I told, lab scale development. It's nothing but a small trials. Now I know what's my tablet weight, what is my hardness, what is my dissolution. I have my drug substance, I have my excipients. Now I will start making the trials. For start with the pre-formulation studies. You might have studied this. Pre-formulation studies is nothing but characterization of the API. Whatever I told before, it's also a part of pre-formulation. Additionally, we will do some compatibility studies, whether the API and this particular excipient, if it is compatible or not, and we will try to look into it. And we will start with some small trials, whether I can go for a wet granulation process, I can go for a roll compaction process, whether I can go for a direct blending and compression process. So we will do some trials and we will try to assess it. And parallelly, not only the formulation development, analytical method development is also equally important. So whatever we are manufacturing, that has to be analyzed right, like how we are doing the small trial and error, the same way will be done for the analysis method also. So that also has to be developed and validated. And there are many ways which we can do this trial and error method, like design of experiments. We can follow the QBD principles, and with the help of some DOE sophisticated softwares we can do, or sometimes we can go for this WOFAT method, which is nothing but one factors at a time. We call it's a simple like changing the binder concentration, changing the diluent concentration, or changing the disintegrant concentration. So it's on a trial basis. So obviously it's a very long process, and not only that, once we come to know that okay, this formula is working for me, and then we will put that into the stability, and as per ICH we will do the stability studies. So for the drug product which is going to intended to be stored at room temperature, the accelerated condition is 40 degrees and 75% RH. And if it is for a US market, then the real time will be 25 degrees Celsius and 60% RH. So we have a dedicated stability chambers for these conditions, and we will load the sample into it, and we will study it for the process of 2 to 3 months, and we will do a trend analysis against the reference product. We will also load the reference product for stability, and we will compare like how it is behaving. Whether sometimes the product may behave initially well, but when it comes to the stability, there may be some trouble. The dissolution may drop, or the related substance may increase. So like this, there will be a lot of challenges. So those things has to be considered, and this is a core development. So whatever the literature says, and whatever we found out in the phase 1, whether it is working or not, that we need to do it here. So this is how lab scale development goes on. Initial and the stability study, and then once everything goes well, then we will say yes, this is my prototype which is finalized. So this is basically a prototype finalization stage. And this is a core of any prototype development, which every formulator should assess initially. The first thing is the quality target product profile. As I told, what is my target profile? It includes my size, shape, hardness, dissolution. What is my target? The name itself says it's a target. So what is my target? You should fix that. And what are all the critical quality attributes which impacts this QTPP? That also you should assess. Like the PSD of the API, it's my CQA, which will directly affect my dissolution. So I should monitor the PSD of the API. I should evaluate the PSD in the sense the particle size distribution of my API, so that my dissolution will be affected. So like that, you should map your CQA against your quality target product profile, and you should work. For that, we will do a risk assessment, linking the material attributes and the process parameters to the drug product CQA. As I told, particle size of the API linked to the dissolution. So the impact is high there. That is what we call it as a risk assessment. The PSD is not going to impact the related substance. So where the risk of PSD with respect to the related substance is low, but the risk of PSD with respect to the dissolution is high. So my concentration will be in finding the right PSD, so that I can map the right dissolution. So that we should list down all my CQA against all my material attributes, and I should give a risk assessment. Normally, the risk assessment will be given in three stages, like low, medium, and high. This is a theoretical assumption. So once we're done with the risk assessment, then based on the risk assessment, we will start working. Our ultimate aim is to bring all our high and medium risk, whatever we have identified, to the low risk. So we should concentrate on this, and we should do the trial and error. When I say my prototype is ready, that means all the high and the medium risk, whichever I identified in this risk assessment, has been reduced to low. So this is what it is. And then based on this design space, which is nothing but design phase and control strategy or interlinked. This will be very helpful when we go for product lifecycle management. Like initially we will have very little bit knowledge on the design space. Like once we get into this product, once we use with this product, then we will have a lot of knowledge about this product. So we can define the control strategy is nothing but, suppose if I say the hardness of tablet is 50 newtons, it's very difficult. Each and every tablet is 50 newtons, correct? So there will be a range. So 50 newtons plus or minus 20 newtons. So it can be from 30 newtons to 70 newtons. I can give it in a range. So I cannot go below 30 and I cannot go below 70. If it goes below 30, what happens? If it goes above 70, what happens? That is my risk, which I have evaluated already during my prototype development. So that is my control strategy, which I need to propose for each and every parameters. Not only that, disintegration time, not more than 15 minutes or 10 minutes, something like that. And then my dissolution, like not not less than 75% at the 45 minutes, something like that. So all those things comes in the control strategy. And product lifecycle management and continuous improvements is the last stage of any product, where once it went into the production, once you get done more and more of batches, where you have more insight on this product, whether we need to able to do any changes. That is what is called as a product lifecycle management. So this is what all about the phase 2, where you will be doing a prototype development, considering all those things. Students, please make a note of all these terms, which is very, very helpful. And also, some people may ask during the time of interviews also. So just to make a note of it. And the third stage comes is the scale up. So as I told, in the prototype development, what we are doing is a small trial and error with some 500 or 1000 tablets max. So what we will do is, there is always a scalability factor. Like when we are doing at a smaller scale, everything looks good, no issues, fine. But what happens when I scale it up? So this scale up is nothing but my internal laboratory. Example, if I wanted to talk about my Axeline, whatever we have a facility to do 500 grams also up to 10 kg. So all my prototype, I have done it in around the 500 grams or 1 kg. So once everything is done, now I will up to 10 kg, which is 10x of my prototype batch. So even at this level itself, what is my scalability? Whether I am able to get the same response, whatever I got for the prototype or not, I need to check. Especially when it comes to a wet granulation, the granulation time, binder addition time, the kneading time, especially, it all impacts your dissolution profile. So all the scalability factors with respect to the particular product, I need to look into it, and whether it is working or not, I need to check at a lab scale up. And again, we will do the stability studies for this lab scale up also. This lab scale up will be maybe around 1 or 2 batches. So again, we will do the stability studies just to, and we will compare it against our prototype batches also with the reference product, whether any impact of the scale up is having any stability or not. So once this is done, then at this phase, we will freeze the formula and freeze the manufacturing process. Also, the analytical method also will be validated and everything will be done. Now we have a product which can be, whatever I spoken is now with respect to our laboratory conditions. Now we are finalized, like this is my product, and now we can go for another scale, another large scale in a production manufacturing facility, which is the fourth stage, which is the scale up. I say it's a larger batch size in a CGMP production facility. Before executing a validation batch, there is a difference between a validation batch and this batch. In general, validation batch, what is meant by validation batches, or some called as an exhibit batch, which is nothing but a batches which we need to make it in the GMP production, and the data of those batches will be a part of our dossier filing, which will be submitted to the regulatory agency. So it is called as a validation or a exhibit batches. So before I execute a validation batches, I am just transferring my product, whichever I have developed in my facility, to a entirely different production facility. So it is not only a scale up. Like for an example, if I done it in 10 kg here, I may go up to 50 kg or 75 kg batch size there, or maximum 100 kg there. It is not only the scale up, it is again a difference in the facility, difference in the instruments. Even though I can say like here also rapid mixer granulator, there also rapid mixer granulator, here also Wooster coater, there also Wooster coater, but still there will be a slight difference. So in order to study the impact of those scalability and the difference in this equipment, we will again go for a scale up batches before we go into a validation batches. Because if anything we wanted to ensure, normally it is not, it all depends on the product's criticality. Sometimes we can skip this batch and we can directly go for a validation or exhibit batch also. But it again depends. But still, I am just telling you people, just for you to understand that in this way also we can go. So again, we will scale it up to a production facility, and all those studies, whatever we have done it here in the scale, we will study it, and we will study the impact of the scalability also. Even if you wanted to do any minor tweaking here and there before we go to a validation batch, we can take it up here. So this is what it is. And this is the batches like which is used, executed for the purpose of regulatory submission. And also we will use this particular batches for a bioequivalence study. And the stability of it will be done as per the ICH, which will be there up to, in general, up to 36 months. Some people may do up to 24 months, but in general, it is up to the 36 months as the real-time conditions. So the basic aim of all those phase 1, 2, 3, 4 is not to have any trouble in this phase 5. The phase 5 should move smoothly because it's a regulatory submission batch. We should not have any surprise at the time of this regulatory submission batch, because we are going to take it in a GMP plant, and there are a lot of costs involved, and a lot of manpower, everything, time consuming process, everything is involved. So nothing should happen with this regulatory submission batches exhibit especially in this exhibit batches or the validation batches. So if you do all those 4 phases properly, and you come to a conclusion that this is what my process is, then phase 5 will be very, very peaceful. So this is what it is. So the next thing is the bioequivalence study. Like I told you, we are a generic developer. So always we should show our product should be biologically equivalent to that particular innovator product. So for that, we will do a bioequivalence study with respect to the reference product. And in some cases, I think you people are aware about this BCS classification, like class 1, class 2, class 3, and class 4. So in some cases, only for the BCS classification, BCS class 1 and 3 molecule, which all falls under BCS class 1 and class 3 molecules, we can get a bio waiver approach, in the sense like without doing a bio study, with the help of some dissolution mapping itself, we can able to claim it to be biologically equivalent to that of a reference product. So that is also there. So please refer to that bio waiver and the BE study guidelines, so you will have that more insight on it. So once the BE study is done, once it is passed, then we can say yes, our generic product is bioequivalent to the reference product. Now we have the product, and we are ready for the regulatory submissions. So this is all about the product development, the various phases, whatever we do, right from the literature search to the bioequivalence study. So I hope I have just conveyed. Actually, it's a very broad thing. Whatever I told is just in a superficial manner, which is just the basic of what we do in the industry. So what are the regulatory requirements? Certain basic things like for USA and Europe, I just compared considering the regulated markets. We should have 3 batches. I told you, that validation or the exhibit batches, we should make 3 batches with a 1 lakh unit or 1/10 of the commercial batch size for both US and Europe. And batches should be of 1 lakh units. That's what I told you people, like 1 lakh units of minimum 1 lakh units is required to be manufactured at the exhibit batch stage. So you just think when anything happens at this stage, and the implications will be very high. So considering this only, all those 4 phases, whatever I told, is very, very critical, and we need to monitor such that these 1 lakh units, whenever we manufacture for a validation batch, is going very smoothly. So this is what it is. And also the stability at the control room temperature is 2560, accelerated is 3075, intermediate. It's all ICH based. So when we are going for a filing, like as I told, it's a 36 month, but we will not wait for the 36 months. We will file it to that particular regulatory agency with the 6 month accelerated conditions and the CRT conditions. Pack, as I said, in USA it is mostly a bottle pack, and in Europe we have both blister and bottle pack. In USA, some products will have some blisters also, but then again, it all depends upon the reference product. So this is all something basic regulatory requirements. Apart from this, there are many things has been listed, but this is the basic things which we need to know. And the last, I have just given some guidelines, which is very, very important. Please go through this ICH Q8, which talks about the complete product development right from the start. Whatever I told, it's all based on this ICH. There will not be any phases will not be given, but actually they will talk about what are all the things to be done. And we will prepare a report called as a product development report, which is the very heart of any filing. So this product development report, it's nothing but like how we started and how we ended, how we finalized the formula. Should have the complete history along with the necessary data should be substantiated in that particular report, which will be a part of our regulatory filing. So this ICHQ8 defines how a product development report should be there. Of course, the stability studies, whatever we wanted to do, that ICHQ and A will define it. And if you wanted to know about this reference product search, as I told you in the beginning, you can go through the Orange Book. It's all free sites. If you put the drug name, you will come to know about the reference product. Especially in US, they will mention it as RLD, but in case of UK and Australia and Canada, they will not mention. But then we need to find it out. I have given one called as MHRA PAR. PAR is nothing but Public Assessment Report. So it is also available for many generics. So you have to just type in Google, for example, X drug public assessment report, you will get it. And then if you download and if you read it carefully, there they will mention what is the reference product they have used and from where they have sourced. So this is also one way of finding the reference product, especially for the UK, Europe, Australia, and Canada. But in US, it is straightforward that in the Orange Book they will listed it as RLD. So this is what I just wanted to cover with you people, and I hope I have just given some basics, which will be very helpful, and you will come to know like what basically we are doing it in our industry basically. So thanks a lot for your time, and once again I thanks the management for organizing this seminar. Yeah. Yes.
Yeah. Thank you. It is the basic of M-Pharm.
Yeah.
What is happening to product development?
Yeah.
Different stages. It is very. I think students.
Yeah.
They are asking you rate.
Yeah. And even whatever I said, it's just in an overview. So there are a lot of things which is there inside that, but still, if they come to know at the initial stage itself, especially when they're going for an interview, even if they refer and if they can able to articulate themselves in what are all the points I said, it will be really a value addition for them.
Any students' questions?
So we have one question from one of our participants.
Yeah.
So other than conventional products, what is the current status of novel drug delivery product development in pharma industries?
Yeah, it's a good question actually. But the novel drug delivery system, it's currently, it is picking up. Almost, if you say the generic product development with respect to the tablets or capsules, it's almost done. Even in Excellent also, we are trying to bring a separate facility for the novel drug delivery systems. There are a lot of scopes involved in it. Yes, now it is slowly coming up. Opportunities are more.
Okay, sir. Thank you, sir.
Yeah.
Bindu Madan, I have some two questions.
Yes, sir.
Hello.
Yeah.
One is you told about this OGD database. Database.
Yeah.
So whether the database will give all the information about the dissolution media, all these things?
Yes. If you go to OGD, it's nothing but Office of Generic Drugs. You can type it in Google, and you have to give the particular drug name. Example, take paracetamol. You just give paracetamol and search. it will give the lot of things like it will give the drug name as paracetamol, dosage form, and also the type of dosage form. For immediate release, what is the dissolution media I should select? What is the apparatus, whether I should go for paddle or a basket? What is the RPM I should select? What are all the time point I should select? Everything will be there. So if we are developing for a US market, then we should follow that particular media only.
Okay. For Indian market?
Indian market, there is no particular database as of now. If it is official in IP, then you can follow that IP monograph. Monograph will say. Even in some cases in US also in OGD, if you say, they will straight away given as refer to USP. That means you have to refer the USP monograph of that particular dosage form. In that it will be defined.
Okay. And from your talk, almost all the institutions, whatever we are developing, we are in the phase 2 only, because whenever a student do a project, we are doing only the pre-formulation. We are making a lab scale product, and we are doing the stability studies. So we are not able to come to phase 3 or the scale up. So we should have some collaboration with the industries to have phase 3. Only these academic projects can be transformed to an industrial project. So later the lab scale and the CGMP, your companies can take off. Am I right?
Yeah, correct. That is correct.
Because as such, in academic institution, we can go with the phase 2 maximum.
Yes.
Product development, phase 2, handing over the basic data of the lab scale of formulation as well as stability to the company. Am I right?
Correct. Yes, correct. Actually, the scale up is very, very important from an industrial point of view.
Yeah, okay. And the container and closure, whether the phase 2 itself, we have to finalize the container and closure, or when we go for scale up, it is okay.
No. In phase 2 itself, we should finalize the container and closure, because after phase 2, there should not be any changes, because the stability, whatever I told, it should be done in our finalized pack. Any change in pack will impact the product quality.
Okay, so the stability study should be carried out with respect to the container and closure at the phase 2 itself.
Yes, right? Once we get the reference product, we will characterize it and we will purchase the same kind of container, and right from the trial 1 itself, we will start using the pack, same pack only.
Okay. And
because
whether we have to go for
if we get into too many variables, suppose if you are changing the pack and the data is failing, we will not know whether it is really because of the product or because of the pack. So we should, the variability should be very, very minimal. Like my pack is the same, but even after scale up, it is failing in the sense that it is a product behavior, not of the pack.
And whether we have to go for a stability study up to 360 days. In academics, it is not possible. So in phase 2, how much we can go for stability studies?
Minimum 1 month, sir. At least 30 days.
30 days. You have to do 30 days minimum stability.
If you have time for your thesis submission, then you can go up to, actually, if you talk about industry, we will call it as based on the product criticality, we will have up to 3 months at a phase 2.
Okay.
So without that, we will not go out with the scale-up batches. But from academic point of view, minimum 30 days is my suggestion.
Okay. Industry point of view, how much?
30 days, 1 month.
The industry point of view?
3 months. Minimum 3 months at a phase 2.
Okay. Okay. Thank you, Bindu Madhavan. If there is no questions, just the organizer will finish off. And thanks for sparing your time.
Thank you, sir.
For the student.
Thank you. Yeah, thank you.
Thank you, sir.
Yeah,
thank you for sharing your knowledge in your field of expertise with us this morning.
Yeah, thank you.
I'm sure the delegates have amassed a great deal of knowledge from your talks, sir. Thank you so much. I also thank the delegates for your participation in this webinar. I thank all those who work behind the scenes to make this webinar a grand success. Thank you, one and all. Have a wonderful Wednesday.
Thank you, sir.